Well, it's great being here. Thanks a lot. I mean, first of all, and it's really great seeing so many people coming to that. And I think particularly, I mean, with Brittany, we have been interacting quite a bit, so it's very nice. It's particularly also because we rarely get the chance to present in this kind of context our work. And also trying to really commit or show a little bit of what we want to do, which way we want to do it. It's not necessarily always the classical works. I mean, Beata works a lot in this area, but we are pretty much in lab interest in epigenetics, development, metabolism overall. And so really this is an opportunity to connect. And the talk before also I liked a lot. I'm kind of seeing where things move forward and what is possible. And we have not yet looked into this database, but I think Beata already took the notes, and so we definitely will get back to that. And basically what I want to do today is I want to give you a bit of an overview. I think there's a lot of been talking, or I think will also be in this, what are treatment options, what is there. We are really basic biology. We're interested in mechanisms. We try to understand what is happening. And Beata will talk a bit about, we try to build up new models where we can test new interventions, new treatment options, but at the same of that, also trying to better understand what are potential ways, mechanistically, the biology behind of that, which may give us an idea of if we see something. You think of newborn screening, think of many of these ways, we also need to know what we are looking for. And to some degree, that's a limitation at this point. You can look for enzymatic changes, you can look at metabolites that change, but we believe, and that's where the epigenetic side comes in, and you will see why we believe this is relevant, these may be new types of diagnostics we can use in maybe in the future, so use in a very early phase to find patients who might be affected. So what I'm showing you here is actually an early embryon and an embryo, very early in development. It's colored here by the level of DNA methylation it has. And so that's basically the epigenetic side. And I'll introduce you what I mean with that and how this links to the one carbon metabolism and how in the overall the developing part comes in here. And maybe because that's also not very clear, obviously we have several layers of information. I mean, all of you are very well aware of the genetics part. For the diseases to develop, the children or those who are affected, they normally have somewhere in the genome a small change, a mutation, and that is a driver of what happens. But at the end of the day, identical twins, which are genetically absolutely identical, and I'm not sure if there's any here, but those who are, they still are not the same person. They are different. They have some difference in them, and somehow that must happen. And so many of the things also, if you have, and I think Brittany very nice showed that or explained that, catching a potential disease early on in life and how this develops, there's a lot of change at which point we start with treatment, what we do to it. So the whole, at the end, the person, how this person presents and what it could be, there's many things that go beyond the genetics. Not pure, I mean, the genome is not the only determinant of what will come out. It's obviously very important, but not everything. And that's a little bit where I want to bring in the epigenetics. So the idea, oops, I jumped ahead one. We can be genetically identical, or with one genetic blueprint, we can create many, many different phenotypes. And that's where the epigenome comes in there. And so there's the idea here that there's basically a second layer sitting on our DNA, which gets modified, that during development, it has to be built up. You have to think about many cell types, and I'll get a bit to that, and how they're instructed, that will later on be relevant for their functionality. And if I very quickly go through it, every one of us has started at some point in our life as a single cell, the fertilized oocyte. So basically the egg cell, which gets fertilized to the sperm. And so at this point, there's this one genetic information which every other cell in our body carries, and these cells divide. And you see here the process, this one cell divides, you have 2 cells, 4 cells, and initially it's a symmetric division, and eventually it changes, and you build up many of these cells. And every time a cell divides, this genetic information is copied, is duplicated, and every other daughter cell will have the same information. And now you have to ask, how is it actually possible now that having the same genetic information, you will have a cell which becomes a brain cell, a liver cell, a blood cell, whatever type. And that's where now this epigenome comes in. So in the end, everybody of us, roughly 30 trillion cells in ours, and roughly 300 cell types. And these, each of them have their own function and their own way they work. And if you now think of the epigenome, this is really a process, and it's a little bit very simplistic here put on there, but it kind of refers to how these cellular changes, the cellular information is stored and how it can be transmitted. And I basically put in here heritable states of gene activity. So how a cell expresses, they're heritable. They can give this from one cell to the next cell, modify that, and that way keep it, but they do not change the DNA sequence. So whatever you got at the beginning of your life as a genetic information, every cell will have, but every cell may use in a slightly different manner. And a brief vignette here. So we have now ways to do that. And one part particularly I want to talk about is so-called DNA methylation. So we have the DNA. This is 4 bases that many of you will hear of, A, T, C, G. And in one position, the C, the cytosine, the body can add small extra molecules, so-called methylation. And methylation, probably in some way, maybe you think of many of the inborn errors, you may have heard that of metabolism, that methylation may have come up several times. And that's actually where I want to get in the end. But this methylation, I'm jumping ahead here one step. So basically can add to it, and you see there, there's these small groups. Is this visible? Oh, no, it doesn't show. Doesn't matter. You see here, basically, to the DNA, we can stick them. Not every position will get that, but where they are put on, they have a functionality. Now, normally, they change how that gene, that region of the genome can be used. And I'm talking now about the genome. And so very quickly giving you that. If you think of our genome, it's at the end like a book. So the cell at the beginning of life has in it a book. It's roughly 3 billion letters. So it's a very long book. Unfortunately, this book doesn't have punctuation. It doesn't have a clear structure. There's no words we can read. So over the last couple of years, we have learned actually to better understand that. And there's the Human Genome Project, which ended around the 2000s. That really gave us an idea, what is there? We kind of, the first time we're able to read the whole book. Now we have this book. Now we've started understanding where are individual parts in it. And so that, as far as we have come so far, and quite well. And so, but we obviously, if you even could read it, let's assume this is a manual to build up a bookshelf. I always like the IKEA resemblance. You go to an IKEA store, you get all of your different manuals, you put them all together. But if you want now to build up the chair or the table, you will need a different part of that. And to keep that, now of this large manual, you will need to know which one actually should I read. If you read the whole thing, you probably don't know how to build up your bookshelf. I'm not sure you basically could stop reading it all. And so if you think of that, ideally, if you would not have it, you would actually mark the parts that are relevant. If you think of a cell, a liver cell will need to know this part of this manual I need all the time. And you can do that. And actually, the cells do the same. With this epigenetic information, which is sitting on there, we can now label specific parts of the genome, this big book, in a specific cell type. A liver cell will have a different type of marking. Different parts of the genome will be labeled. Parts of, let's say, we should use it. These things are very important. And this one, don't even look at it. We don't need it. And so really, you can use it. And so that's really now allows being kind of the epigenetic part. You put on top of the genetic information. You can keep it. It's heritable. So once the cell divides, all the daughter cells will keep that information too. So a liver cell, or kind of a precursor of a liver cell, once you make more liver cells, they will still remain liver cells. They will not suddenly become a neuron, and the other one a blood cell. They will stay what they are. And so this allows now the cell to take this DNA, the genetic information, transcribe it into something which is called RNA, messenger information, and this one can then be used to make the proteins that we need at the end to make the system work, make the cells work themselves. And so just to very quickly jump back here, this one last step is, so we now have the genetic information, and in every cell we can label part of the genome. That's the part we need, that's on. Other parts we tell off, and we do it in a cell-to-cell type. And so now we have this one manual. We can make multiple different types of epigenomes, many multiple types of genetic use of the information. What also is very important, and I think that's where, if we now start thinking about how can we modulate it, if you think about life, what happened in the past, is it forever there? This is not a steady state. This actually is a dynamic process. Because now we have that information, which is written on the genome. On the one hand, we need those proteins, these molecules, we can put it on there, they write them. We can also erase it. And ultimately, if you then want to use it, you think of the IKEA manual again, you being the reader, you can also be kind of know which part to read. And so that really is now the way how we convey that information. So far now talked a bit about methylation, or brought it up a few times. And just to illustrate, this DNA methylation is indeed very dynamic. And this is now what we did here a long time ago. We looked at how does the DNA methylation in a cell change. This is just looking at the total levels. If you look at how much DNA methylation, how many of these sites which can have this modification on the change over time. And you find here, there's the oocytes, so the egg cell and the sperm. They have different levels. Once they come together, we form the fertilized egg cell. We call it a zygote. And that in the early stages, it actually loses a lot of methylation. There's a kind of a process that we call reprogramming. So the body wants to remove it to then be able to build up the new person. And that really happens here. It is at the very early stage where we have the stem cells. And Beate will later briefly come also to this concept of stem cells, which are pluripotent. They can make any other cell type in the body. And they then start gaining a lot of methylation. They develop, and all of the somatic tissues that we're interested in will be developed in that process. But you can see already, on the one hand, it's a very dynamic process. But as you see here, you also need a lot of methylation. You need a lot of these molecules to be added to the genome to maintain that information. And that's a little bit the point where obviously it's relevant also for the body to have appropriate amounts of these molecules to then be added to really instruct the cell. It's like if you want, again, coming back to my manual, if you want to label the parts of your manual that you will need, but you run out of ink, at the end you will end up with a manual which is not properly labeled, where you don't know which information you want to keep. And so here, indeed, also nutrition comes as quite a key part. So really the diet that we have is one of the sources that we need to supply the body with this, the so-called ink, or the metabolites that are now put on the genome to be written. And you will already see here that we have folic acid, methionine, producing SAM, the S-adenomethionine, that really is then being used to add these methyl groups to the DNA to then store that information long term. And so really, these nutrients really build the building blocks what we need. Folate, vitamin B12, choline, betaine, many of those. And I think in some of those, you will actually be aware, you know, we need to add it to diet. Some of them are even in treatments that are being used to really rescue or kind of support development. And here comes now in the one-carbon metabolism, in the one-carbon cycle. And that is a really central part also in metabolism and overall biology, because it really builds many of the building blocks. Not every part that the body needs, not every of the molecules has to be supplied by the diet, but the body has then to use those, to modify them, and at the end use them. And I mean, obviously, we have the folate cycle, the methionine cycle, and these two are really key integrated in the whole process of early developmental biology. On the one hand, we have this part, which is the DNNTP synthesis. So these are the building blocks for making new DNA. Every time a cell divides, as I before said, this DNA, this genome information has to be amplified, and both daughter cells need that. We need 30 trillion cells. So from the beginning cell, 30 trillion cells have to be made from that one. And that means we need a lot of the building blocks that are there, and we need this whole cycle to actually generate them. So if we have a defect in that cycle, that already will be a significant implication and have the implication of actually making that new. So cell division, amplification of the DNA will be limited. And then the other part, which is directly links in, is this methionine cycle, where here you see the DNA methylation. So again, providing the parts that we now need to modify the DNA to add this extra epigenetic layer on top of it, in addition to other methylation pathways that we have also in the body. And then some other part, which I'm not going to touch on here, this transsulfuration pathway. And so obviously this means if now we have any inhibition, any kind of changes to that pathway, if they don't work properly, we have, on the one hand, some other metabolic consequences, but particular nuclear biosynthesis and DNA and histone epigenetic changes are very likely to happen. And Beate will, in the next part, come a bit closer to that. If you think of that, there's particularly a couple of enzymes which are so-called rate-limiting enzymes. If these don't work properly, this whole cycle gets out of sync, it doesn't properly work, and so we need to think about why don't they happen. But there is a part which is obviously very relevant. And I don't show it here, but it's shown here. These pathways, they normally don't have only a single entry point. And that's, I think, also where the part you can think of, if many of the treatments we have, if you have a change somewhere in here, you obviously could try to feed the cycle at other parts. You can kind of think of ways how metabolites, nutrients can be used, or which parts we need to replace. And so if you have a change in the activity, for example, in the MTHFR gene, that will result in change in metatetrahydrofolate changes, but also the direct link in the methionine cycle, and so will affect all of this part. And so we need to think, can we potentially feed other parts, methionine? Is that a way to overcome change we have over here, or which way can be modulated? And so as I think going stepwise through there, we can better understand what happens. One limitation we still have from the biology is that wherever we are tissue-wise, it's very challenging sometimes to provide these metabolites. In one part, if you now start thinking of the Homocystinuria, are that obviously we have no defects on some of these enzymes, and as such, in production of these metabolites, SAM particular, that we then later need for methylation. And unfortunately, we cannot easily constitute it because the brain particular is kind of in a privileged zone in the body. It's protected from the rest of the body. Metabolically, the blood circulation has to go through the blood-brain barrier. So transporting anything into that site has limitations. And so we need to think about how can we overcome it. Can we with precursors, with other ways, or maybe also with just dosage, high dosage, can that change? And I don't really want to go now into all of the clinical symptoms. I think Britain's database that they're building up will much more reflect and also the reality which is there, not just the expected one. But what we want to do, and that's where Beata will come in, is think about how can we actually address potential things. What can we actually contribute to the whole thing? Because we are not clinicians, we are basic research. We try to understand the mechanisms, but ultimately then translate that. And so where we really want to move, and I think we're doing progress, and Beata will go into that, is can we better understand the biological reasoning? Why actually does a change in the activity of these enzymes lead to change that we observe? I think you have now some phenotypic data of what you see, what the patients present with, but how does that actually, what is the biology basis for that? And so if you have a growth change, why? I mean, is it just because there's no nucleotides? Where does the methylation system come in? Can we affect that? Can we look what happens? And by developing novel models, because at the end, we would want to have something that is as close as possible to our patient population, which is as good as possible representing what is the reality. I mean, there's animal models which people are used, but they not necessarily always fully reflect that. And at the same time, we need a system which is scalable, which we can test many different interventions, which we can look at very early processes, because many things are probably happening. Like you said, if you wait 11 years, a lot of changes will have happened. So how can we know what is the best point of addressing that? What do we need to know? How do we need to do that? And so here, really, can we develop novel approaches? And Beata will talk a bit about that. And then really then with the intention to test novel approaches also. In the second part, and I only added that recently, and it's not really what we're doing that, but not part at the moment of the main projects, we're quite interested in monitoring and detection. And that is because this methylation system, as I said, is very relevant in the whole body. And we work on newborn screening. We are interested in the blood, looking how methylation in the blood changes with the expectation that if there's a reduced or there's not sufficient methylation there, this likely will affect methylation in many cell types and like in the blood. And this may be in very early proxy to get a feeling of what is happening. Can we use that diagnostically? We have good methodology to very early screen that. Beata's done actually can generate whole genome methylation maps from single blood drops. And so the potential is there. We kind of mostly actually limited here in having proper patient samples with, and I think that's what I mentioned before, so with control samples, ideally siblings, genetically very similar but not identical. So we can actually look what is now this one small change happening. And so really this would be very excited also discussed about on the databases, the kind of the registers you're building up to have access to that. And with that actually I will stop. My main goal was really giving you an idea of the area where we are, why we believe this is an important area that the epigenetics may or likely does also play a role in here, and how we can integrate it in the treatment long term. And I think I can hand directly over to Beata, which I also prefer not to say the last name because I'm also not sure if I always say it correctly. Thank you very much.
Thanks a lot, Fernand. Thanks a lot all for having me here. I will briefly give you an idea of what I've been working on and specifically on the progress that we've made on establishing the cortical organoids that have deficiencies in a number of remethylation disorder genes. So Fernand already showed you the cycle. So essentially, I think one key thing to highlight still is that, well, we are looking at remethylation disorders, which is essentially this impaired transition from homocysteine to methionine. And there's many genes that are involved in these disorders, but in our purpose, we decided to focus on a few. So particularly methionine synthase, because it directly affects the pathway of interest. So it would give us the clearest kind of view of what happens when that is impaired. Okay, can you hear me? Okay. And then two others that are the most common in remethylation disorders, so MTHFR and MMACHC. And then we also included CBS because it sort of acts in our purpose as a control. in a sense that it also has high homocysteine, but it doesn't have decrease in SAM, or at least we don't expect to have a decrease in SAM. And as Ferdinand mentioned, basically neurodevelopment is one of the key affected processes in one-carbon metabolism, and especially when it's impaired. But it's something that is very difficult to model, and that's the reason for our project, where we're trying to establish an organoid model. So what is an organoid? So it's essentially 3D ball of cells, very small, it's in millimeters in diameter. But if you were to take it and slice it up, look under the microscope, you would see folds and shapes and cell types similar to that in early brain development. So that's very cool because you can use it in various applications. Starting from basic research, you can use it in testing various treatments, assess things like gene expression, metabolites, electrophysiology, etc. And it starts one step back. Basically, to build these organoids, you need to have cells. And in order to build model of the disease, you need the diseased cells. There's two ways to do it. You can either take the patient cells and reprogram them, or you can take the healthy cells and also reprogram them and then introduce the mutation. So we decided to go with the route of healthy cells because then we could have isogenic disease cells. And how it works, we basically get the fibroblasts, so skin cells from healthy individual. These then get reprogrammed to iPSCs, which is essentially the cell type then that under the right conditions can become basically any cell in the body. So it's very fascinating that you can model it to become anything. And essentially then we create an environment that would mimic the brain development. So we tell the cells be brain-like, and then it becomes this 3D structure organoid that starts forming these folds, starts layering with progenitors, neurons, etc. So once we had the healthy cells, we had to introduce the variants that also are present in patients. And we do this using now very well-known system CRISPR-Cas9. The way it works, it's sort of molecular scissors that we can guide it to a very specific area in the genome, and it will go and it will cut, and it will produce variants of all kinds. And then basically we're able to screen that at bulk population to see whether any edit took place at all. And once we see that that did indeed happen, we can screen individual clones. We do that by looking at the sequence. So basically, since we target a sequence of interest, we can see whether there's any mutations. So in this case, I'm just showing a couple of examples of variants that are sort of unchanged and changed, so you can see that the edit took place. But obviously, just because there's a variant or a polymorphism, let's say, it doesn't necessarily mean that the function would be impaired or lost. So we have to confirm that actually the function is impaired. So in the case of MTR, for example, we use a formate incorporation assay. So this is an assay used in clinics and diagnostics on a regular basis, where we feed the cells with radio-labeled formates and measure out the methionine amount. And we can see basically if there's no incorporation, methionine synthase is inactive. And we indeed find that. So we find a lot of clones that do lose the function. And we select 3 in the end for statistical purposes and 1 unedited. So we really have the control that goes through the same process as all of our knockout lines and remain as wild type, we call it. So sort of healthy wild type, you'll hear me say interchangeably. And we do the same for MTHFR. So for MTHFR, there is a direct assay where basically we can directly measure the activity of MTHFR. And we also find the clones that both lose the function and remain as wild type, so we can have this very elegant, controlled system for our models. Now, the editing is a very stressful procedure for the cells. iPSCs, induced pluripotent stem cells, they don't like to be cloned, edited, manipulated. So we have to make sure that after the process, they do remain stem cell-like. And we do that through a few different quality control steps. So one being we check that they still express the stem cell marker. So you can see it's a immunocytochemistry staining. So basically we can take the cells and label different proteins and have these beautiful color pictures. And in this particular case, you can see SOX2, SSCA4, NANOG. These are canonical stem cell markers, so the cells should express that. Similarly, KS67, it's an indicator of proliferation because the cells are known to proliferate, and that's what you want with the stem cells. And so they all do express that. Another quality control step that we do is we look at whether the cells actually can differentiate. So you do want them to be able to differentiate to all stem cell lineages or all lineages, yes. And those being ectoderm, mesoderm, endoderm. And we again check that all cells do that. And we also check that by editing, we don't introduce any genomic abnormalities. So in this case, you can see you want the copy number of all these different spots to be around 2. That means that there's 2 chromosomes. And because it's a male donor, you can see that the Y and X, they're 1 of each. So basically, we confirm that the cells are of good quality and they're stem cell-like. And then we can take these cells and continue with a differentiation to the organoids. And this is a well-established protocol. We just make some additions, but essentially we take the cells from stem cells. We allow them to form these 3D balls that we call embryoid bodies. We induce neural lineage. We allow them to expand and differentiate. And then through a long process of growth and maturity, we keep them until 90 days, but you could actually keep them indefinitely and allow them to grow and mature. Basically, people keep it for months at a time. And then we can compare the healthy and the diseased states at different time points. One additional thing that we do to the original protocols, original methods, is that we add the different methionine conditions because methionine is a key player in remethylation disorders. So you'll hear me refer to low and high, or sometimes I will change to the values. So the high is 200 micromolar. That's essentially what is used in standard protocols, but it's way, way, way above physiological. And low, we do 5 micromolar because that's something that would be very low, yet still normal in a healthy human being. And so these would be the first results. So I will show you basically some of the genes, and I will mention others when relevant. But so in this case, I'm showing you MTR, but the same thing we're seeing in MTHFR and MMA-CHC. So here you can see the organoids on day 7, 12, and 16. And here it's the wild type, and here's the knockout line. And there's 2 conditions, so low and high. So you can see in the wild type, there's not much difference in size. There's some decrease in size in the low methionine condition, but it's not as obvious. However, you can see that overall methionine knockout organoids are overall much smaller, and they kind of stay smaller. And the difference between low and high is quite significant. What's interesting, though, is that over time, so here you can see organoids on day 70, the difference in the wild type completely disappears. So the wild type methionine low, methionine high kind of catch up. Same with high methionine and MTR knockouts, they do catch up over time. But the low methionine one stays small. So they do grow, but if you compare this line to the rest, it's quite a bit smaller. And we see that also in MTHFR and MMA-CHC. Now, I tried to quantify it, so you can see here the growth over the 90 days. And here you can see the high methionine, and you have the low methionine condition. And in black, it's the wild type, and in blue is the CBS, which we actually weren't sure what we would expect to see. And interestingly, it seems like in low methionine condition, it's also not affected, might even be slightly improved at an earlier stage as it grows a bit more. But as you can see, MTHFR, MTR, MMA-CHC, they're all significantly smaller in low methionine conditions, but high methionine conditions seem to not have a strong effect overall, let's say. And then this is another way of quantifying this. So this is at different time points, so not over time. And here it's interesting in that basically the stars above indicate how significant the difference is, and NS would be non-significant. So if you take an earlier time point, day 18 to 23, each dot here is an organoid, and blue is high, and blue is low, and green is high, methionine. At earlier time points, there seems to be a difference in all conditions, including the wild type and CBS. However, as the time progresses, wild type and CBS, the difference between high and low methionine becomes insignificant, and it sort of stays that way until the end. But you can see that in all the remethylation defects, so MTR, MTHFR, MACHC, the difference only gets bigger over time, and it doesn't catch up, even though the high methionine conditions, the size, as you can see in green, it's quite comparable across. So then we wanted to see, basically, is it really that the size is different, or are they just going in different paths? Because it could be that maybe the high methionine, standard methionine conditions, the organoids develop as they should, but then the small ones, they become something else. So we did some earlier organoid staining. So this is basically, you take an organoid, you slice it up, as I mentioned, and then we can stain it for different markers. So in this case, I'm showing you an example of MTR knockout, high, low, and looking at neuronal markers. So SOX2 is a progenitor, early progenitor marker. MAP2 would be the neuron marker, so it's sort of later mature neuron. And as you can see, both conditions do have the markers there. I think you can appreciate the size difference. So the scale is the same, and it's basically the full organoid. This one almost doesn't fit in the picture. But it seems like the cell types are the same or similar. Similarly, we wanted to see whether the expected structures are there in both conditions. And we do see that. So here I circled some because it's a bit difficult to see. So you do expect to see these rosette formations. Sometimes it's difficult to catch them because depending on how you slice the organoid, you might slice it kind of in a wrong angle so you don't see the rosette. But we do find them in both conditions. And FOXG1 here is also a marker of forebrain. So it's basically the region that would develop into cortex. So we also can confirm that both conditions end up going in the same region of an organoid or sort of the brain. And another way to quantify it or to look at this is through looking at gene expression. So here I'm showing a few markers, and here it's different genotypes grouped together, and day 90 high and day 90 low, and in gray it's the day 0, which would be basically IPS state. So what we expect to see is that all the neuronal markers go up. So here would be the progenitors and then kind of later progenitors into neurons, and we expect them to go up from day 0 to day 90, which they do in both low and high methionine. And we expect that they go into the forebrain region, which they do. So here, again, you see both in the high and low methionine, they are higher than day 0. And similarly, we don't expect to see any posterior brain regions developing. So that would be kind of the spinal cord and so on. And here we don't see that. So essentially, it seems like the methionine depletion doesn't affect the cell type populations or regionalization, at least at this sort of high-level assessment. However, we still don't fully understand what the effect is, so we're doing further analysis to really quantify and understand it. So at this point, we can see that high or low methionine does have an impact on how the organoid grows, and there does seem to be impaired growth in remethylation disorder conditions, which is not the case in the healthy or CBS-affected organoids. But we don't know whether that translates to function. We don't know if that's really, if cell type population and composition is the same, because the methods that I showed you, they're not perfectly quantifiable, and they're all assessing the bulk population. So what we're doing next, we're basically looking at the single-cell nuclear RNA-seq, which is essentially a method that allows us to individually sequence all the genes expressed in each cell, so we can pinpoint what RNA come from which cell, and with that we can explain basically how this organoid is composed and whether the similar cell types behave in a similar way. We also are looking at electrophysiology. So this is a method that allows us to see how the organoids actually function and what kind of electrophysiological activity they have, and whether it's different between the two conditions. We also plan to look at some DNA methylation to see whether this lack of SAM or no lack of SAM, this is also something that we want to check, affects anything in particular on DNA level. And yes, so SAM-SH ratio is something that has been in the field referred to as methylation potential metric. So we are measuring that to see whether different conditions and also different genotypes have any changes in the SAM-SH ratio. And that brings me to my conclusion. So I think at this point, the data is very preliminary. So I would suggest to take that with a grain of salt, but it does seem that methionine does play a role in growth, possibly not fate, but the time and the next results will show. Something of interest is also that the effect might be state dependent. So there does seem to be a window of sensitivity. So possibly if we were to reintroduce methionine at some point, perhaps that could reverse the growth defect, but it's hard to tell without actually experimentally testing that. There does seem to be a genotype-specific effect. So as I mentioned, we don't see the effect in wild type or the CBS knockout lines, which suggest that it's really remethylation disorders that are sensitive to methionine effects. And overall, we are happy that we have the screening platform, which can be expanded to further compounds to be tested. It can be used to also test different time points. In theory, it could also be expanded to different organoid systems. So something of interest could be retinal organoids, because we know that the eye symptoms are quite severe. But yeah, we have these cell lines that have the mutations and can be used, and we're happy to collaborate and expand and do more things. And with that, I would like to acknowledge all the people that participated in this work and all of you for your attention and for all the great work that you do, and happy to answer anything.